Hey everyone! Welcome to today's webinar hosted by Biochina. In our China for Global session, we'll be covering the latest technical and industrial developments within the Chinese bioindustry, as well as exploring international business opportunities. For today, we have a webinar titled Mouse Models of Metabolic Diseases and Their Applications, from Disease Research to Drug Development. If you're looking to enter the Chinese market and connect with key decision-makers at Biochina, don't miss out. Use promo code BIOP0926 to get 20% off your partnering registration before October 31st. Click the banner to register now and secure your spot. Also, be sure to follow us on LinkedIn to stay updated on the Chinese bioindustry and get the latest news about our upcoming events. Let's now welcome our speaker, the scientist Jinhua Ye from GemPharmatech.
Hello, everyone. Welcome to GemPharmatech webinar. And so my topic today is mouse models of metabolic disease and their applications, from disease research to drug development. I'm Jinhua Ye. And the global burden of metabolic and cardiovascular disease is very heavy. And as we can see from this picture, the heart disease and diabetes are among the top 10 causes of death worldwide. And this information is from the World Health Organization. And these two diseases are accounting for 10 million deaths in 2021. On our metabolic and cardiovascular platform, we have mouse models for many different diseases, including obesity, diabetes, dyslipidemia, gout, liver injury, MASH, and cardiovascular disease. And especially today, I will talk about the DBD mice and the DB plus NOT3 knockout mice for diabetes. And I will focus on the AMS1 knockout mice for the MASH model. And on the right, it's a list of some humanized mouse models, which are quite useful for efficacy study of short interfering RNA medicine and antibody-based medicine, if the antibody only recognizes the human protein instead of the mouse protein. Especially, I will talk about our new humanized GLP-1 receptor mice and the humanized GIP receptor mice for the obesity study. And I will also talk about the humanized AGT mice, which is a very good model for the hypertension study. Besides our offer of mouse models, we also provide multi-platform support for the clinical research. We have our platform include cell engineering platform, in vivo efficacy of a drug study platform, a phenotyping platform, flow cytometry, and histology analysis platform. So with the help of this platform, we can complete our clients' projects successfully. The content of my talk today will have 4 parts. So first, obesity. I will talk about the humanized GLP-1 receptor mice and the humanized GIP receptor mice. Secondly, diabetes. I will talk about the DBDB plus NOT3 knockout mice. And then about liver disease, I will focus on the AMS1 knockout mice, which is a very good model. And lastly, in the 4th part, I will talk about the hypertension, and I will focus on the humanized AGT mice with the AAV H-renin transduction model. So first, obesity. So GemPharmatech has plenty obesity research model. We have the normal standard diet-induced obesity model. We also have monogenic mutation model, like the OB mice and the DBDB mice. And we also have the AMS1 knockout mice. These mice develop obesity as well. So in the diet-induced obesity model, we offer standard B6 mice, and we also can induce obesity in our humanized GLP-1 receptor mice and our humanized GIP receptor mice. And on these models, we have tested many drugs. For example, we test semaglutide and tirzepatide on the humanized GLP-1 receptor mice, which I will talk about it later. And on the humanized GIP receptor mice, we tested a long-acting GIP, and we also tested a drug. The name is GMA107, which is a fusion of human GIP receptor antibody and GLP-1 receptor agonist. So first, about the humanized GLP-1 receptor mice. When we make the mice, we collect tissue samples, the brain and the lung. So by qPCR, we can detect the human GLP-1 receptor expression in the humanized mice. But in normal mice, we didn't detect the human gene expression. And in normal B6 mice, we can detect, of course, the mouse GLP-1 receptor expression, but we don't detect the human GLP-1 receptor expression. And we also collected the pancreas tissue. So by staining, we can see here the human GLP-1 receptor is expressed strongly here. And we cannot detect any signal of the human receptor in normal mice. So this data prove that our construction of the humanized mice is successful. After we get this mice, we use high-fat diet to induce obesity on the humanized mice. So we can see the green one, it's the humanized mice fed by the high-fat diet. So its blood glucose increased, but after the treatment of semaglutide and tirzepatide, its blood glucose decreased. And also after the glucose tolerance, if the mice is fed with a high-fat diet, its glucose tolerance is impaired. But after the treatment of the semaglutide and the tirzepatide, its glucose tolerance is improved a lot. And also, after the high-fat diet induction, the cholesterol rise in the blood. But after the treatment of the tirzepatide and the semaglutide, the cholesterol level decreased. So it is the same with the triglyceride and the LDL cholesterol. So the semaglutide and the tirzepatide treatment can improve the hyperlipidemia induced by the high-fat diet. And then we also tested the GSP. The GSP is a diagnostic marker of the diabetes. After the treatment of the semaglutide and the tirzepatide, the GSP level decreased. And also the fat tissue weight is also reduced after the treatment of semaglutide and tirzepatide, as you can see from these two charts. So this is the I have talked about the GLP-1 receptor humanized mice. Then this is the humanized GIP receptor mice. After we constructed the mice, the same way as the last mice, we collected tissue samples. So in the brain and the fat tissue, we can, in both male and female mice, we can detect the human GLP-1 receptor expression. But we cannot. But in the normal B6 mice, we do not detect the human receptor expression. And in the humanized mice, we barely detect the mouse gene expression. But in normal B6 mice, we can detect the mouse GIP receptor expression. So this data proves that our construction of the humanized GIP receptor mice is successful. And after we get this mice, we do a drug efficacy study. We give the mice the DAGIP. So the DAGIP is a long-acting GIP analog, as we can see from this picture. So in normal mice, after we give the normal B6 mice with the GIP and with the DIGIP, the glucose tolerance is enhanced. And if we give the DIGP to the humanized GIP receptor mice, it has similar result. So it shows that the human GIP receptor works well in the humanized mice. And then we also collected the brain from the mice. And it's because based on literature, we know that the DIGP controls the food intake through the central neuron system, and it will activate the expression of the C-force gene in the central nervous system, especially the hypothalamic feeding center. As we can see from the staining, after the dosing of the DIGP, the C-force gene is activated in the normal B6 mice. And similarly, in our B6 humanized GIP receptor mice, we get the same result. So these results together prove that the human GIP receptor protein works normally in our humanized mice. And then we test a drug, GMA107, which is a human GIP receptor antibody and fused with GLP-1 receptor agonist. And we use a mouse GIP receptor antibody as control. So we can see after we feed the humanized mice with high-fat diet, its body weight increased a lot. And if we treat the mice with the human GIP receptor antibody, the body weight of the mice decreased, but the negative control of the mouse GIP receptor antibody does not have this effect. And we get a similar result on the liver weight and the cholesterol level. So this proves that only the antibody that targeting the human GIP receptor can functions well in our humanized mice. If it is a mouse GIP receptor antibody, it cannot bind it to the human GIP receptor in our humanized mice, and it does not have the body reducing effect. Second about diabetes. So we have several different types of diabetes research models. For type 2 diabetes, we have the normal DBDB mice, which is widely used. We also have this overnutrition chemical injury model. And for type 1 diabetes, we have the NOD mice, which is an autoimmune model. And we also have this chemimal injury model. So the MLDS means multiple low dose STZ. And for the type 2 diabetes, we also have the DIO mice, which is a model of overnutrition. And we also have the AMS1 knockout mouse model. Especially today, I will talk about the DBDB plus NOD3 knockout mouse model, which is a very good model for diabetic nephropathy study. So this model combines the leptin mutation with the INNAS knockout. So in this paper, the authors compare many different types of diabetic nephropathy mouse models, and they conclude that the endothelial NAS deficiency, which is another name of the NAS3, is a very successful strategy for enhancing kidney injury in a mouse model of diabetes. So we believe that the DBDB plus the in-NAS knockout mice is a very good model for the diabetic nephropathy. And from our data, we also get the same conclusion. So as we can see here, the DBDB plus NAS3 knockout mice has heavier body weight than normal BKS mice, although its body weight is not as heavy as the DBDB mice, which is the blue line. And for the glucose level, the DBDB mice and the DBDB plus NAS3 knockout mice shows similar level of blood glucose, which is much higher than normal mice. Especially for the UACR, the DBDB plus NAS3 knockout mice shows much higher level of UACR than normal DBDB mice and normal BKS mice. So the level of the UACR for the DBDB plus NAS3 mice is almost like 4,000, but for DBDB mice and the normal mice, it's quite low. And also from histopathological changes, we also see very obvious phenotype. So the DBDB plus NAS3 knockout mice shows very severe phenotype in glomerulus, basophilia, inflation, interstitial fibrosis, and the tubule protein. Especially when they are like 12 weeks old. As we can see, the red one is the DBDB plus NAS3 knockout mice, and the black one and the blue one is normal mice and the normal DB mice. So from these results, we conclude that the DBDB plus NAS3 mice could recapitulate the feature of renal lesion of clinic patients at different stages, including proteinuria, glomerulus injury, tubular basophilia, tubule protein, inflation, and fibrosis. Then we do a drug efficacy study on these mice. We test the semaglutide and the lisinopril, which is an anti-hypertension medicine. So from the first picture, we can see that the single use of semaglutide or the lisinopril. So the semaglutide is the green line, the yellow one is the lisinopril. They can indeed reduce the UACR, but if we combine these two medicines together, they show better results. So this means that the combination use of these two medicines can improve the proteinuria phenotype. And for the HbA1c, the single use of semaglutide has very good result, but the lisinopril does not have this effect. And for the glucose, it's the same. The semaglutide, the green one, shows very good effect, but the yellow line, the lisinopril, does not have this effect. So we conclude that the semaglutide can improve the hyperglycemia in this DBDB plus NAS3 knockout mice. And also we collect the kidney. We do stainings. So from the H&E staining, we can see that the semaglutide and lisinopril can partially rescue the renal lesions. especially for the tuber protein parameter. As you can see here, the green one is the semaglutide, and the yellow one is the lisinopril. Compared to the black one, the control one, the tuber protein score is reduced, and the combination use can further rescue the tuber protein phenotype. And also for the glomerulus, single use of semaglutide and the lisinopril can improve the glomerulus injury, and the combination use have a better effect. Then the third chapter is the liver disease. On liver disease, especially for the MASH, we have many different types of models. We have the high-fat diet and the Western diet induced model, which is caused by overnutrition. We also have the nutrition deficiency diet induced model, including the MASLD and the CDA-HFD. We also have the overnutrition plus chemical injury model, the HFD plus carbon tetrachloride, and the HFD plus STZ injury. We also have the OBOB plus Western diet model, and especially we also have a monogenetic model, including the DBDB mice and the ARMS1 knockout MASH model. And today I will focus on the ARMS1 knockout MASH model. For other liver injury model, we also have the chemical injury model, like alcohol, and we also have the surgery model, like the bioduct ligation model. So before I move on to the ARMS1 knockout mice, I want to talk a little bit about the Astrom syndrome. So the Astrom syndrome is a rare monogenic recessive multi-organ disorder. It's caused by the mutation, homozygous mutation of the ARMS1 gene. And its phenotype include childhood onset obesity, type 2 diabetes, vision defect, sensory defects, and other complications. And the function of the ARMS1 protein is supposed to function in the intracellular trafficking to the primary cilia. As we can see from this cartoon, this blue box is the ARMS1 protein. It's located on the cilia for transport, and it's also the ARMS1 protein is also supposed to be involved in the vesicle transport from the Golgi apparatus to the cilia. So we can see it here. This is the ARMS1 protein. It's involved in the transport. And then, based on this literature information, we made the ARMS1 knockout mice. And after we made the mice, we find that these mice indeed develop obesity, hyperglycemia, dyslipidemia, steatohepatitis, and fibrosis. So these mice develop this phenotype since so when we feed these mice with chow diet, it can develop these phenotypes after like 8 weeks. They develop mild fibrosis after about 8 weeks of feeding of the chow diet. And after like 12 to 14 weeks of chow diet feeding, they develop moderate fibrosis. So the time is a little bit long. And if we want to accelerate this progress of the disease, we can use high calorie diet, like high fat diet, to induce the phenotype, to accelerate the phenotype. It will save us time. So this is our data. We can see here that so on chow diet, this green line, the AMS1 knockout mice, its body weight is heavier than the normal mice. But if we feed the AMS1 knockout mice with high-fat diet or Western diet, the body weight increased much faster. And for the blood lipid, like the cholesterol, if the mice is fed with the chow diet, it's a little bit higher than normal mice. But if we feed the mice with high-fat diet or Western diet, the cholesterol level increase a lot. So it is the same with the LDL cholesterol, the plasma ALT, and the plasma AST. These two are the markers for the liver function. And for the glucose metabolism, we can see that the high-fat diet and the Western diet can also increase the blood glucose level. And the high-fat diet and the Western diet also increase the plasma insulin level, which means that they can impair the insulin tolerance. And when we do the glucose tolerance test on the mice fed with different diets, we can also find that if the mice is fed with the high-fat diet, its glucose tolerance is impaired much more severe than mice fed with a normal chow diet or Western diet. And then we collected the liver from the mice. So from the HT staining, we can find that so the high-fat diet and the Western diet can increase the MASH score. So the steatosis, the inflammation, and the ballooning of the liver is worse after the high-fat diet or Western diet feeding. And also we can see from here that after the Western diet feeding, the liver weight to body weight ratio is much higher than control diet or high-fat diet. And also the fibrosis phenotype is also more drastic after the feeding of the high-fat diet and the Western diet, as we can see from this picture. And we also do the alpha smooth muscle actin staining. So from the alpha smooth muscle actin staining, we can see that the high-fat diet and the Western diet increase the SMA signals. So in summary, the AMS1 mice can develop the MASH phenotype if they were fed with the chow diet, but it takes a little bit long time. If we want to accelerate this progress of the disease, we can feed the AMS1 knockout mice with high-fat diet or Western diet. This saves us time, and the phenotype is also more severe. So after we establish this model, we do a drug efficacy study. We test the tirzepatide and the atherosclerosis on the AMS1 knockout mice. This is our study outline. We choose these two medicines because these two medicines show very good results in clinical trials. And indeed, in our results, we can see that after the tirzepatide and the atherosclerosis treatment, the body weight of the AMS1 knockout mice is reduced to a large extent, the yellow line, the blue line, and the yellow line. And the food intake is also reduced. For the glucose, when we fed the mice with so the AMS1 knockout mice will develop hyperglycemia, But if we treat the mice with tirzepatide or the efruxifermin, the blood glucose level is reduced. So it is the same with the triglyceride. The treatment reduced the triglyceride level, and also the cholesterol. The cholesterol level is reduced after the treatment of the two drugs. And it's the same with the HDL cholesterol and the LDL cholesterol. And we also do a blood test to check the liver function. So the ALT and the AST level is reduced after the treatment with the two drugs. We collect the liver. So from this picture, we can see that compared to normal B6 mice, the AMS1 knockout mice, its liver is much bigger and it looks pale. But after the treatment of the two drugs, the liver size becomes normal, and its color turns normal, like brownish. And the liver weight. So from this chart, you can see that the liver weight is reduced after the treatment, and also the triglyceride content in the liver is also reduced. When we do the H staining, and staining, and do the MASH score, we can find that after the treatment of the two drugs, the liver, the fatty liver phenotype is is rescued to a large extent, as we can see from the staining, and the MASH score is also reduced. So it's the same with the fibrosis. So after the treatment, the fibrosis in the liver is reduced. We can also see that from this fibrosis score. So in the last chart, I'll talk about hypertension. So this on the left, this picture shows the regulation system of blood pressure. So this is the standard RAS regulation of the blood pressure. Here we can see the renin, the angiotensinogen. So the renin will convert the angiotensinogen to angiotensin 1, and the ACE will convert the angiotensin 1 to angiotensin 2. And also from this picture, we can see the our aldosterone is also here. It also regulates the blood pressure. And we can see that the renin, the angiotensin 2, the ACE, and the aldosterone are popular targets for anti-hypertension drug development. And the last year, a paper published on the New England Journal of Medicine, a drug, the leberceran, which is an RNA interferon therapy agent. It shows very good results in controlling the blood pressure. And the target of the leberceran is the AGT. So we believe the AGT is a very promising targeting for treat hypertension. So this is the background that we make the humanized AGT mice. And another reason we make the humanized AGT mice is because that it's necessary to humanize the target gene, so as to meet the demand of the efficacy study of the RNA drugs, because the mechanism of action of the RNA interfering drug is based on the complementary base pairing of the short interfering RNA oligo with the messenger RNA in the cell. So when we make the humanized AGT mice, we induce hypertension in this mice by AV transduction of human renin. We can see that after the human renin overexpression, it can indeed induce hypertension in the humanized AGT mice, as we can see from the SBP and the DBP. And we also can see that the AAV human renin transduction does not impair the liver function, as we can see from the ALT level. And we also see that the hypertension in the mice impaired the kidney function and caused albuminuria. So as we can see from the BUN and the creatinine, the BUN and the creatinine level increase, and the UACR level also increase after the hypertension. We also do other analysis. So in the heart, we find that after we induce hypertension in the humanized AGT mice with the AAV-H-renin, the ejection fraction in the heart is decreased, and the fractional shortening is also decreased. And also the heart develop hypertrophy, so we can see the heart weight increase, and the heart weight to body weight ratio is also increased. And at the same time, the heart also developed fibrosis, as we can see from the MASLD staining. The blue area shows where the fibrosis happen. And also the blood vessel also has problems, as we can see that the aortic aneurysm happened here, and also the aorta dissection happened, and also the thoracic aorta wall thickness is also becomes thicker. So in summary, today I talk about four disease, obesity. I talk about the humanized GLP-1 receptor mice and the humanized GIP receptor mice. It's a humanized mice with diet-induced obesity, and we also test 4 drugs on these 2 mice. Secondly, I talk about the diabetes, focused on the DBDB plus 3 knockout mice. This mice is a double knockout mice, and 2 drugs, the semaglutide and the lisinopril, shows very good results on this model. For the MASH, I talked about the RMS1 knockout model, which is a monogenic model, and 2 drugs, the tirzepatide and the efruxifermin, shows good results on this MASH model. So finally, in the last part, I gave an introduction about our hypertension model, the humanized AGT mice with AAV human renin transduction. It shows the typical phenotype of hypertension. So thank you. This is the end of my talk.
Biokina 2025 is happening from March 13th to 15th, bringing together 3,000 Chinese biotech companies and 30,000 participants. If you're looking to enter the Chinese market and connect with key decision makers, this is your opportunity. Use promo code BIOP0926 to get 20% off your partnering registration before October 31st. Click the banner to register and secure your spot now. Also, be sure to follow us on LinkedIn to stay updated on the Chinese bio industry and get the latest news about our upcoming events. See you next time.